The first thing we need to do is prepare the laminar flow hood. Preheat the tool sterilizer by turning it on using a switch in the back. After it's done blinking, press and hold the set button until the red running light is on. Then use 70% ethanol and spray it all over, making sure to get the sides of the hood, the tool rests, and the work surface. Close the hood and turn on the UV light to let it dry for a few minutes. Before entering the hood, spray your hands and arms with ethanol, ensuring to cover any watch or jewelry you are wearing. For tools, you will need different sized forceps, small scissors, the circle cutting tool, and its corresponding stick. Place them into the glass beads of the tool sterilizer and leave them for a couple minutes until they get slightly hot. Bring into the hood the plant jar, the media plate, and a large petri dish. Now that the tools are ready, carefully take them out and place them on the tool rest to cool. While we wait, we can label our petri dish. We will be using a sticker label that corresponds to the genotype of the plant. In this case, we are using the Royal Royce, so we will use a red sticker. Then, using a thin Sharpie, write the genotype of the plant, the type of media that it is plated on, and the date that it is being plated. It is crucial that you never reach over any open petri dishes, containers, or tools. To start, grab an autoclaved large filter paper disc using a sterile forceps and place it into the large petri dish. Next, we will be adding some autoclaved DI water with a sterile pipette to the filter paper disc. This is to keep the plants from drying out as we are working with them. Again, take note that I am never reaching over any open containers or dishes or tools. This is the sterile strawberry donor plant. We will be using some small scissors to cut a leaf at the base of the root. Then you can use some forceps to take it out and place it onto the filter paper disc on the petri dish. Close the jar and place it off to the side. Now we will be preparing our leaf discs, so we will use a combination of forceps in one hand and the circle cutter tool in the other. Hold the leaf in place and use pressure to cut out a circle on the leaf. Repeat this throughout the leaf to get as many circles as possible. What this does is create a wound on the leaf, and this is what allows the calluses, or undifferentiated cells, to form. You can use forceps to help remove the remaining leaf scraps from the leaf discs, which you can set off to the side to discard later. If you have a leaf that is about the size of a circle, you want to still cut it to create the wound to induce the callus. Repeat this for other leaves on this section. If at any point your tool falls onto the table or you touch it, please place it back into the sterilizer for a couple minutes to ensure that there is no contamination. It is better to be safe than sorry. If the leaf discs get stuck into the circle cutter, use the stick and place it through the top to push them out of the bottom, and then you can use forceps to separate them. Once you are done cutting up the circles from the leaves, we will be cutting the petioles into segments to also be plaited. Using scissors or a scalpel, cut it into small segments. The plant scraps can be put into a pile to discard later. Now we will be transferring them to the media plate. Carefully remove the lid and place it off to the side where you will not reach over it. Using forceps, place the leaf discs on the media. Make sure you are placing the top of the leaf or the darker waxy side touching the media, and the lighter side will be facing up. Place them slightly spread out, and it makes it easier if you place them in a pattern such as lines. We will also be plaiting the petiole segments. Repeat this until you are done with all of your segments and leaf discs. Hinge to close the dish and set it off to the side. Repeat this with the remainder of the plant. This is an example of the leaf discs getting stuck in the tool and using the stick to push them out and then separating them using forceps. This is the Preculture plate all done. Carefully hinge to close the lid without reaching over it, and now we will be sealing it using parafilm. Remove the paper backing. Hold the lid tightly shut, and then hold one end of the parafilm and stretch it around, making sure it is sealing the lid onto the petri dish. This is the finished plate. Place your tools back into the sterilizer for a final cleaning and then let them cool down. You can discard the large petri dish with the filter paper and plant scraps. Finally, place your plate and plant into the incubator.